here, and we have talked of protein nanoassembly, and you know the basic process. From DNA, you'll have transcription. The RNA would go to the ribosome, and the ribosome would then translate it into a protein. But I'm sure you would have read this in your in molecular biology classes. So protein nanostructures are ubiquitous in cell and serve as catalyst, structural support element, and molecular recognition. Antigen, antibody, structural support element include actins and tubulins. And of course, catalysts, you have a whole lot of enzymes that work as catalysts. So you know that. These biomicromolecules are assembled through transcription and translation process, that also you know, and which lead to primary, secondary, and tertiary structures and confer function such as catalytic activity. The function is only present in its tertiary or quaternary structure of the protein. It is not present in the primary or secondary structure where it is not completely folded. And then, of course, you can have post-translational modifications which may activate, inactivate, or alter protein function. Nature has thus evolved its own optimized toolkit through evolution that allows a large number of protein nanostructures to be constructed naturally. And now what we are doing is we are taking these designs into making artificial nanostructures that can be of help to us. So protein nanoassembly, inspired by nature's machinery, there has been an emergence of research activities to evolve synthetic strategies that allow rational design to construct functional protein nanostructures. So what we want is a functional protein nanostructure. Our function is predefined, and our structure also is more or less predefined because we want it in a specific range and a specific conformation so that it is functional. Proteins are attractive building blocks for design of functional nanomaterials due to their inherent bioactivity and multiple functionalities. So bioactivity and attractive material properties allow proteins to be the molecules of choice for making nanostructures. They are also stable, biocompatible, and biosafe. However, as I told you, the lack of predictability of how the protein is going to fold and what function it may produce is what is the major limitation. But luckily, as of today, we have enough knowledge base that allows us to, from bioinformatics, from biotechnology, and from chemical biology, that can allow us to reasonably predict that given a primary structure, what is it going to be in terms of its secondary, tertiary, and quaternary structure, and what is the function that it is largely going to take. So now, with the advancement in biotechnology, with the advancement of a lot of prior knowledge, now it is not so much of a limitation. Okay. So this is what you do here. You have neurotoxins, you have actin, which is a polymer of actin filament here, and we can have other structures. What we want to do is to try and bring together two different polypeptides or two different proteins or two different domains. What are domains? Domains are specific regions within the protein that have the functionality. So what we want to do in terms of protein nanoassembly is to try and bring together either two different polypeptides or two different domains or two different proteins, complete proteins together. So here you have a protein A or protein B, or a polypeptide A or a polypeptide B, or a domain A or a domain B. And what we want to do is to bring domain A and B together using what is known as supramolecular linkers. Supramolecular linkers will have affinity to A and to B. So because they are themselves connected to each other, when you bring them close to protein A and B, the two proteins will come in contact, and this may result in a combined function, which could be a combined function of their two individual functions. It could also result in a absolutely novel new function. Then, of course, you can bring, you could have an intermediate molecule here, which is having a supramolecular linker for A and a supramolecular linker for B. And now you have three different proteins coming together, or three different polypeptides coming together, or three different domains coming together. And that domains coming together can again result in a synergistic function, a combined function of all three, or altogether new function. And then you could also have multiple of the same protein, same molecule together, same polypeptide together. That can help you achieve a function, more efficient functionality as compared to what you currently have. So there are several possibilities. Again, in order to form a predefined protein nanostructure, stringent control over directionality and spatial placement of protein building blocks is essential. Therefore, surfaces of protein building blocks should ideally be encoded with molecular information, and that is contain the supramolecular recognition motif as an anchor point. So this is your supramolecular linker. So this must have this A protein or A polypeptide that we want to get in interaction with B. this green one should also have complementarity to your A protein, and there must be something on the molecular surface of A protein that interacts with this green part of the supramolecular linker, allows it to attach to it. Likewise, if you want to bring a B protein or B polypeptide or B domain here, then again, the B domain must have some sort of complementarity or lock and key fitting model or induced fitting model between this B supramolecular linker and the B molecule here, so that they can interact, and now you have the two domains coming together, right? So basically there has to be recognition elements on the surface of the protein which are identified by your supramolecular linker, and therefore now you can have interaction between the protein and your supramolecular linker. At one end it is interacting with A, at the other end interacting with B. Because they are now in space together, they may now have a combined function. That is what is the idea. These can interact with the respective supramolecular linkers in a lock and key like mechanism for a spontaneous generation of distinct higher order protein assemblies. So then, of course, what are these supramolecular linkers? These supramolecular linkers can be of several types. Most commonly, what we use is streptavidin, biotin. This is important because streptavidin and biotin are two molecules that have strong affinity to each other. So if you want to bring two molecules together, coat one with streptavidin and coat the other with biotin, the two molecules stick with each other automatically. Likewise, you could have certain epitopes expressed on your recombinant protein, and you can generate antibodies against epitopes, and this epitope antibody interaction can allow you again to isolate your recombinant protein. You're trying to bring the two proteins, two polypeptides or two domains together. What you can also do is link one of the domain with a particular DNA molecule, single-stranded, and link the other domain with a complementary DNA molecule. Because these are complementary, they will find each other, bind to each other, and because they're binding to each other, they'll bring the two domains close together here, right? And in this case, you can also have azobenzene as one of in the polymer chain. With azobenzene, you can control when they pair and when they do not pair. So when the UV light, when you show them the UV light, they'll unpair and they'll be far apart. And when you show the visible light for 50 nanometer and beyond, they will come together again. And you can see the action when they're together, what is the function they're doing, and when they are separated from each other, what is the function, whether the function is disrupted or not, right? Likewise, what you can do is you can have your DNA nanosheet. So this is your DNA scale DNA sheet here. And on this DNA, you have two strands again. This strand here is complementary to the blue strand on the red protein. This violet strand here is complementary to the black DNA strand on the blue protein. Because these strands are complementary, these two domains that are bound here, or the polypeptides that are bound here, all the proteins that are bound here, would automatically get attracted and get bound onto this DNA nanosheet. So now what you have is basically a compartment where you have two different enzymes or two different polypeptides or two different domains coming together, and now the same can be checked for function again. So DNA itself can be used as a supramolecular linker to arrange multiple proteins into well-defined nanostructures. DNA linker can be directly coupled to protein of interest and then assembled onto a DNA scaffold. Alternatively, the protein of interest is attached directly to the DNA scaffold, and both approaches require either covalent or non-covalent conjugation of the desired protein with the DNA. So what you want is basically your DNA has to be attached to the protein of interest. So this can be done covalently or non-covalently, but the bioconjugation between the DNA and protein has to be there. And then, of course, the bioconjugation between DNA and the DNA sheet has to be there. So all these things are, possible and are being actively tested now, right? Then, of course, what you can also do is you can make a closed compartment. And again, you have three DNA strands here in this compartment, and you have three protein molecules that have complementary DNA strands. So these would automatically come and hybridize here and get immobilized in this compartment here. So you can make, you could have a cascade of proteins of the same pathway in a single compartment, and that could then again help you get a particular, mimic an organelle and gets its reaction going. So this is what you can do. So you can make an artificial mitochondria, or you can make an artificial chloroplast conceptually. These are not being done as of now. These are still under test. But then conceptually, what you can do is you can mimic an organelle, a biological organelle, using this method where you have proteins of the same pathway coming up and in a chamber, that can itself be of a DNA chamber, right? So non-covalent methods include biotin, streptavidin. We have talked of that. Histag, nickel, nitro, low triacetic acid, aptamer interactions. Covalent strategies to connect proteins and DNA as supplying include disulfide and melamide coupling proteins, ligation, bioorthogonal click chemistry, enzyme-mediated reactions. So not going into details of this. DNA nanostructures are beginning to be investigated as tools for drug delivery and similar applications because they provide precisely programmable scaffolds for attachment of functional groups, including drug and targeting moieties. So you could also, if you want to give a drug, a single drug, you could use a DNA nanostructure and you can include a drug here. If you want to give multiple therapy, then again, 3 different drug molecules can be attached into a single covalent structure, a single DNA nanostructure. And then you can also control how this DNA nanostructure is going to open and close, which will come next. So here we talk about how you can have new functionality by changing the domains or exchanging the domains between proteins. So if you see here, this is gene A, it has exon 1 codes for hand, exon 2 codes for full. Likewise, this is gene B coding for awesome. You understand what handful means? Handful means a very small amount. Awesome means, like this lecture is awesome, right? You guys are awesome. So you can then have an exchange of segments here. So you are basically exchanging the exon 2 between these 2 genes. And now what happens is you have altogether new protein, which is handsome. And then you have another protein here where the exchange has happened. This is awful. So these are basically 2 new proteins with an altogether different function. So I'll use the analogy of words. Basically, words have different meaning. Likewise, the proteins that you get after domain swap have altogether different functionality. And this happens also biologically. Most of the new proteins that you see and most of the new functions and novel functionality that you see that have come up in the genome across the evolutionary years has been largely by, gene duplication and also by domain swap.